Hello everyone, my name is Brian, and in today's video I'll be going over the nitrate reduction test, which is exercise 5-6. Alright, first things first, let's meet the microbes. In this lab you'll be dealing with A. faecalis, E. coli, and finally Pseudomonas aeruginosa. And of course Pseudomonas aeruginosa is a BSL-2, so you guys will need to wear gloves for this lab. Alright, so let's just quickly outline what you'll need. As I mentioned earlier, you'll need gloves. You'll also need to grab a tube rack and, of course, prep your Bunsen burner. Alongside that, 4 nitrate broth tubes with the reagents A and B, just one of each. And finally, the zinc powder, which you'll need towards the end of the lab experiment. Now the lab is broken up into 2 parts. In the first part, you'll just need to label your nitrate broth tubes. So starting with the 3 organisms that you are given, just label it with your name, date, and the organisms that you have, and finally for the last one, that will be your control tube. And once you have them all labeled, you will then inoculate, of course excluding the control. And once you have successfully completed inoculating your nitrate broth tubes, you will then just place them in the rack located in the back of the class, and the bioprep team will then incubate them for 48 hours at 37 degrees Celsius. Now, for the second part, you'll get your nitrate broths back, and the first thing you want to do is to check if the bacteria has successfully grown, and this is usually noticeable by looking for any signs of turbidity. Once you've seen that all of your nitrate broths were successfully inoculated, the first thing you want to look for is gas in the Durham tubes, as that is usually the first indicator of whether or not you have a positive or negative result. After you've checked for gas, you will then add 8 drops each of both reagents A and B to all of your nitrate broths. And once you have done that, to then record your first results. Now, after you've added reagents A and B, you'll notice that a few of your tubes will not have changed color at all, and those tubes will then require zinc. And once you have added zinc, you will then notice that there should have been some color change, and you want to record those results again. And at that step, you'll be at the end, and with all of your results recorded, you will then have to interpret those results and write down all of your interpretations in the table located in your lab manual. Alright, so with the overall outline of the experiment discussed, we can then dive into the theory behind the test itself and how it is applied. So the main purpose of the nitrate broth is used to detect the presence of an enzyme known as nitrate reductase. This enzyme allows bacteria to perform a single-step reduction of nitrate to nitrite. In other words, it just basically means that the bacteria is able to convert the nitrate into nitrate. The secondary purpose of this test is to identify the different ways that nitrate can be reduced by bacteria, as the single-step reduction is just one of many ways that bacteria are able to utilize and reduce nitrate. Some can reduce it into molecular nitrogen, while others can produce a whole variety of nitrogenous compounds such as ammonia. Alright, so let's kind of dive into a little bit of how this test works. What are all the different chemicals and mechanisms in place that allow this test to tell us what bacteria you are dealing with? And so for the nitrate broth, obviously it is a medium that is rich in potassium nitrate, which I have used these dark blue molecules to represent as the nitrate within the medium itself. And these purple molecules here as other chemicals and nutrients that are mixed into the medium in order to allow the bacteria to grow successfully. So with that nitrate-rich broth, if you inoculate those tubes with a bacteria that can produce nitrate reductase, that bacteria will then take all that nitrate that is within the medium and break it down, leading to a large buildup of nitrite. Since we can't really see that nitrite in the solution, that is when we start adding reagents A and B, and the role of these reagents is to allow us to be able to confirm that the bacteria was capable of reducing nitrate into nitrite, because the nitrite is important for these reagents to interact and thus work in creating this red color change that is indicative of an organism that is positive for nitrate reductase. So just to kind of talk about the chemical interactions real quick, as I mentioned earlier, the nitrite in the medium will then form another compound known as nitrous acid. That nitrous acid will then react with sulfonyllic acid, which is reagent A. And that reaction will then form another compound known as diacetized sulfonyllic acid. As the concentration of diacetized sulfonyllic acid increases within the medium, it will then react with reagent B, known as naphthylamine, which then creates that final product known as p-sulfobenzene azo-A naphthylamine, which has a red color, and that is what develops within the medium and changes the color from a more turbid gray into a bright red color. Alright, so for you visually minded folks out there, I've created this diagram to kind of help visualize what is going on in that molecular level, where all of these chemical reactions are occurring that leads to that red, that leads to the development of that red color. So if we were to kind of zoom into this medium that you had just inoculated with some bacteria, you'll have noticed that this bacteria was capable of producing that all-important enzyme that was able to convert that dark blue molecule nitrate into nitrite, which is represented by these light blue molecules here. And finally, at this step, you then added reagents A and B, reagent A being sulfonyllic acid and reagent B being naphthylamine. And what you'll notice here is that the sulfonyllic acid is represented by this light purple molecule, and the naphthylamine is represented by this light green molecule here. And now you have in this solution a lot of sulfonyllic acid molecules interacting with the nitrate or nitrous acid within the medium. And as those two chemicals interact, it then forms what I mentioned earlier as diacetized sulfonyllic acid, which is then capable of reacting with naphthylamine, and finally, after this reaction, will then form p-sulfobenzene that is responsible for the generation of that red color. Alright, so after adding reagents A and B, you'll notice that a few of your tubes will not have changed color at all. So what is going on here? So in that situation, it can mean one of two things. it can either mean that, one, the organism was not able to reduce nitrate into nitrite because it isn't capable of producing nitrate reductase, or the organism was able to break down nitrate into molecular nitrogen or some other nitrogenous compound. And I apologize here, it should be nitrate. And because it was able to further break down nitrate into molecular nitrogen or ammonia, for example, those are all chemicals that are not capable of interacting with reagents A and B. Thus, it explains why you don't see a color change here. Now, the next step in this experiment is to be able to figure out which one of these is the correct scenario within the broth itself. So how do we figure that out? That will then require the powdered zinc to be added to the medium. And once you add zinc to the medium, if that medium is full of nitrate, that zinc will then be able to reduce the nitrate into nitrite. And once there is nitrite, since we've already added reagents A and B to the solution, it will then be able to interact and cause that chain reaction of chemical reactions that lead to the buildup of p-sulfobenzene that produces that bright red color. All right, so once again, another visual diagram to take a look at. As we can see here, these dark blue molecules that represent nitrate is mixed into the solution alongside our reagents A and B. And this jar here contains our powdered zinc, and what you want to do is just take a very small amount of that powdered zinc and just pour it into the test tube. And in this diagram now, you can then see the zinc molecules within the solution. And those zinc molecules, what they do is they reduce that nitrate into nitrite that then forms nitrous acid. And as I mentioned earlier, it is nitrous acid that really kickstarts everything. Reacts with sulfonylic acid, producing the diazotized sulfonylic acid. And finally, the interaction with naphthylamine will then create our final product that produces the red color. All right, so what does all of this mean? First of all, after adding the zinc and noticing a color change, it tells you that the organism is not capable of producing nitrate reductase. That is because after adding reagents A and B to the medium, there was no color change that occurred because you need nitrite in order for those reagents to produce that color. And the color that was produced was not because of the organism itself, it was because of zinc. Since zinc has a capability of reducing nitrate. And once the nitrate is reduced into nitrite, it is then able to react with reagents A and B. And thus, it tells you that the organism is negative for nitrate reductase. Now finally, there's going to be a broth medium where there is no color change at all, even after adding zinc. And in that situation, it tells you that the organism has reduced the nitrate into some other nitrogenous compound or even molecular nitrogen. And those are all not able to interact with reagents A and B. However, it still shows you that the bacteria is capable of reducing nitrate and thus is considered a positive result, even though there is no color change. Alright, so towards the end, we'll go over the interpretation guide. Now what you see here is the pre-zinc phase and the post-zinc phase. So for those of you who may have caught on, you'll have noticed that in the first phase that the red color change tells you that the organism is positive. But after adding zinc, the red color change is negative. And I know for some students, and it has caused some confusion for me, and it is something that students tend to slip up on. And it is due to that sort of complex situation where where some organisms are capable of just producing nitrite, while there are other types of bacterial organisms that, one, are not capable of reducing nitrate, or as mentioned already, that the organism mostly reduces it into some other nitrogenous compound, and those nitrogenous compounds do not interact with the reagents that lead to that color change. And so when I approach the nitrate test, I like to split it up into two halves, just so I know that when I see the red color change, I'll be able to distinguish whether the color change in that situation is considered positive, while in the other situation it should be considered as a negative result. And so that is why I have this dichotomy here, where in the first phase it is the pre-zinc phase, and that is obviously the phase where zinc has not been added, and the post-zinc phase, which is where the zinc has been introduced into the medium. And of course, in the pre-zinc phase, the red color change is positive. And as I've talked about it already, that is because the organism itself was capable of reducing the nitrate into nitrite, and thus the availability of nitrite within the medium allowed those reagents A and B to then interact and form that color change. And after that, you will have noticed that there will be two of your test tubes where there was no color change, and that is when the addition of zinc is required in order to differentiate whether or not, one, the organism was totally incapable of producing nitrate reductase, or if the organism just broke it down into some other nitrogenous compound that is unable to react with reagents A and B. And in this situation, when you do see that red color change, you know it wasn't because of the bacteria itself, it was because of the zinc. And thus, because it was the zinc that reduced the nitrate into nitrite, that in that situation, that organism is considered negative, since it is unable to reduce nitrate. But if there is no color change at all, even after adding reagents A and B and the zinc, the color still remains the same. That is considered positive, and because that bacteria was able to reduce the nitrate into some entirely different nitrogen compound. Alright, and that brings us to the end of our video. Thank you so much for listening. Once again, if you like this video, please like it and leave me a comment. I'm currently open to suggestions. I'm trying to experiment and try out a lot of different things to sort of help explain these labs through an online format. So your suggestions will be much appreciated. Thanks again, guys, and have a good night.